Hello, everyone. My name is Omar Santizo, part of Belmont's research group. So today I'll be talking about my research called Improving Undergraduate Chemistry Education, Analyzing Biochemical Redox Analytes Using a Student-Built Potential Stat. So an overview of my presentation today will be our purpose, the TNC stat, improving education, biochemical and electrochemical education, implementing into the curriculum, build a portfolio, results, future work, conclusion, references, and acknowledgments. So the purpose of the research was to modifying existing equipment typically used by the maker community with a vision to bring a low-cost, accessible instrument into the undergraduate lab. Bring in biochemical perspectives that happen in the human body that relate with the reagent used in the experiment. So what is this device that we buy into the research called? So the device is called a TNC stat. So the TNC stat is basically a potential stat. As you guys know, potential stats are very expensive. They run from 7,000 to 20,000 dollars, which makes it very expensive, you know. So undergraduate institutions such as Dominguez, Cal State Fullerton, and in some cases UC Irvine makes it hard for them to buy this instrument. So we decided to make a low cost potential stat for under $30. So what is this potential stat made of? So the potential stat is made of three components: the breadboard, the TI LMP 91000, the TZ 3.2 control, the TI LMP 91000 is an amplifier circuit that is used to convert raw data into digital data. The TZ 3.2 controls a hub, a place where all the data is gathered, that communicates with the LMP to give the user flexible data for analysis. And then you guys might say, oh, what type of graphs or data this TNC that performs? It performs selective tomograms. So selective tomograms is a method using analytical chemistry for students to understand or professor and so on to understand about electrochemistry movements of electrons. So then three selective tomograms are given in three samples, and they are called reversible, irreversible, and quasi-reversible redox reactions. So how can the thesis stat improve undergraduate education? The thesis stat can improve undergraduate education by allowing the students to build their own thesis stat. And then this will introduce the field of electrical engineering. This will also tie into Belmont's lecture of discussion of concentration. As Professor Belmont's lecture this concentration, the students will be able to understand how, as analytical concentration increases, single detection by device increases. So they will also understand the method of selective photometry, which is part of Professor Belmont's uh, course, which is called quantum analysis. So the students will also be able to correlate real life applications with the reagent they will use for the experiment and so on. And finally, the team stat will be able to bring in the understanding of redox reactions, which is the movement of electrons. And then, as you guys know, students at Casa de Hills, they only learn this concept once or twice in general chemistry. So our vision was to bring in this experiment for them to understand the concept in a deeper level, and so they could take it with them for graduate school or any upper division course. So my part of the experiment was to build a portfolio with the different analytes. So I worked with acerbic acid, acetaminophen, and acetylcysteine. So let's talk about acerbic acid. So you guys know acerbic acid is vitamin C, and you guys could consume vitamin C with strawberries, oranges, and so on. So acerbic acid is an antioxidant. It's used to kill free radicals in tree survey, and this will demonstrate an irreversible redox reaction. So the way we're gonna correlate acerbic acid to real life is by demonstrating and explaining what's a free radical in our body, and then how the free radical can express a gene in a certain part of the organ, which then cause a disease, for example, cancer. So as you guys see in the picture on the left, free radicals are caused by alcohols, fried foods, tobacco, pesticides, and so on. So then if the student is able to consume vitamin C in oranges and so on. The antioxidant, which is vitamin C, will donate its electron, and then the free radical will be neutralized, which then will be killed, and then the immune system will deal with the rest, which will then create it into waste in urine or in feces. So this is the way we're gonna correlate it and how movements of electrons happen in the body as well. So now let's talk about acetaminophen. Acetaminophen is also known as Tylenol, is a pain and fever relief. It demonstrates a quasi-reversible reaction. As of right now, we do not know um, how we will correlate it to the real life. However, our vision is to correlate with headaches. So that will be our vision and our work for future to come. So N-acetylcysteine is another antioxidant um, which prevents asthma attacks, and can treat rashes and other skin problems. And the way we're going to correlate it with acetaminophen is that doctors today use N-acetylcysteine for overdose of acetaminophen. So the N-acetylcysteine will work as an antioxidant, which then neutralize the acetaminophen and will prevent any damage to the liver and make the patient feel much better after they receive treatment for the poison of overdosing of acetaminophen. And then acetaminophen will also illustrate quasi-reversible cellular biochemistry. So now that we uh we move on, I want to compare the results of our potential stat to a real potential stat. So before we go on, so we know is there's a reversible reduction reaction on black, quasi-reversible reaction in blue, irreversible quasi irreversible redox reaction in red. So irreversible is the simultaneous redox to oxidation. So the movement of electrons happen pretty fast right after another. So the peak down here is reduction. The peak on top is oxidation. And for quasi is similar. However, it takes longer from the electrons to move. So the you guys see is it's like it's getting longer, so it takes a little bit while for it to happen. And irreversible reaction, uh, ascorbic acid is the one that demonstrates the irreversible, and then it oxidizes pretty fast, so it would just show the oxidation peak. So now let's compare it. So ferrocyanide, it shows a reversible redox reaction. So then if you guys compare this one with the black one, similar, very similar. And then ascorbic acid, so this is irreversible. We compare it with the red one, it's very, very similar. So now let's compare it with acetaminophen. So acetaminophen is the quasi-reversible and N-acetylcysteine. So then if we bring it back to the blue one, it's quasi-reversible. So it's very similar as well. So then calibration curve. So the way we saw that our TC stat was reliable is if our R square value was higher than 0.98, our TC stat is very reliable. So for if it passed the test for ferrocyanide, passed the test for uric acid, passed the test for N-acetylcysteine and acetaminophen. So our device is very reliable. So implementing into the curriculum. So to implement the curriculum, in 2019, we actually performed an experiment with a group of students that were ready for any extra credit that Professor Belmont offers. So we broke it down into two lab periods. The first one was demonstration. The second one was for them to do the standards and solutions and run the experiment. And these are the students' results. So they were able to get they were working with first cyanide. They were able to get a reversible redox reaction. However, the R square value was a little bit lower than our hit value, which is above 0.98. However, we found this as a success due to being the first trial for the students. So future work. We're planning to complete the experiment for the students and bring everything together so they can start working on it and have students conduct the experiment as time comes, as COVID leaves and stuff. So they will start doing the experiment. And lastly, keep testing the tendencies that with any other analytes such as glucose. So this is a continuation of future work. It will also improve their laboratory techniques. It will improve their understanding of cellular photometry and biochemical and electrochemical applications and so on. So conclusion, it's has been a success. Belmont Research Group has developed a thesis stat, and it's low cost, so it's affordable for us and for the students to understand, silicon photometry, electrochemistry, how movements of electrons happen in the body, and so on. So acknowledgement, I want to acknowledge the McNair Scholar Program in the Office of Undergraduate Research at CSUDH. Special thanks to our two mentors, which is Barbara Belmont, Justin Hathaway, and my partner Claire Down. And then highlights from 2019 to 2021, me and my partner won first place at an oral presentation and a poster presentation. And Justin Hathaway won third place at Student Research Day as well. So this is our references, and thank you for coming, and thank you. Hope you guys liked it.